Oh. So now in this session, in this class, we will see the animated form of DNA replication. We will understand how the DNA is actually constructed, designed, and how the polymerization and how the replication of DNA actually is happening. We have been studying and पढ़ी आसुचे ए भिडियोटारे आमे केवल we will understand the animation of the total process, whatever we have been studying in our previous classes, those things only I will just show you in the animated form to develop the imagination level about this concept. So see, this is the DNA, double stranded DNA, how it is looking like. These are the nucleotides. Each nucleotide have, see, pentose sugar, the nitrogenous base and the phosphate group. Okay, how the nitrogenous base cytosine, guanine, thymine, adenine are complementary base pairing. There are 3 hydrogen bonds between G and C and 2 hydrogen bond between A and T. See, these are 2 opposite polynucleotide chain of a double stranded DNA. The gote hela 5 prime to 3 prime end, the another is 3 prime to 5 prime end. See, 5 prime to 3 prime, 3 prime to 5. Both are anti-parallel to each other. These are anti-parallel really. Both strands and the opposite nucleotides are complementary base pairing according to the nitrogenous base. See the animation part. So DNA विषय idea हला, then we will see this is how the semi-conservative nature of DNA, माने the parent strand is actually separated, the new strand is developed. See how the new strand is developing to form 2 different DNA. Therefore it is called as semi-conservative nature of DNA. We studied that. That देखिला हमें parent strand किमिति separate है कि नया strand तैयारी कला. Now we will see the process of replication mechanism हमें देखिवा, where the DNA starts unwinding, that is called as origin of replication. We will see that. See, this is the origin of replication where the double stranded DNA starts unwinding. Okay, unwind kala. So unwind and both direction replication haba, that is called replication fork. See, this is the complex, all the enzymes, all the replication complex, means whatever the enzymes we have studied, the topoisomerase. I would make a note. Topoisomerase is also called as gyrase, G-Y-R-A-S-E, important. Make a note, lekha, notes re lekha, and amaro jo printed material diya hai se pakhare madhya lekha, topoisomerase pakhare, topoisomerase bracket lekha or gyrase, G-Y-R-A-S-E, DNA gyrase boli madhya kohchanti. So topoisomerase is also called as DNA gyrase. Agode jinsa I have given in assignment re mu deichi DNA clamp boli. A clamp da actually topoisomerase pakhre bi rahuchi plus DNA polymerase pakhre rahuchi. Karana what is the need of clamp? Clamp is used to hold the 2 strand of DNA perfectly, parallelly. दीटार DNA रे दीटा स्ट्रैंड को से होल्ड कर रखिबो. तो टोपोआइसोमरेज पूरा मध्य क्लैंपिंग सिस्टम अछि एंड DNA पॉलीमरेज रे मध्य क्लैंपिंग क्लैंप जान कि कोडा लोड होल्ड कर रखि आपनी क्लैंप. तो एमजे अछि this thing I have given in assignment question. Assignment रे गोटे कॉलम मैचिंग रे मुसे क्वेश्चन टा देछि. Anyway, come to the point here. So we will see how different enzymes perform, how the gyrase, the helicase, the primase, the polymerase, the ligase. These are the enzymes, how they are performing during the whole process of replication we will see. We have studied these things in details, but now we will see the process here. See, the strand is already, the origin of replication, it is already unwind, already separated. Now the new strands are actually formed. Okay, new strands are actually formed. These are template strand and these are newly synthesized strand. This is helicase. Okay, helicase unhelix the DNA, and this is the gyrase or topoisomerase, and the single stranded protein we have seen that how they bind. This is DNA gyrase or topoisomerase. We know the helicase and gyrase helps in unwinding or unzipping, unzip or unwind the double stranded DNA जे कि unhelix करिवो तार hydrogen bond को breakage करेगी. Then single stranded binding protein will come and prevent the separated DNA strand for rejoining हमारे prevent करिवो सी. Okay, then see, this is helicase, this is topoisomerase and single stranded bond. This is DNA polymerase. This have clamp, see, beta clamp loader, means the clamp is actually holding the both DNA polymerase on the both strand, and the total DNA strand को hold करिवो plus DNA polymerase को मध्य hold करिवो, because parallelly leading and lagging strand synthesis हवार अछि, continuous and discontinuous strand synthesis हवार अछि. आमे पढिचे एवडा सब तो how parallelly हवार अछि से क्लाम्प लोडर दरकार. एठे देख कौन हला प्राइमर आसिकी बाइंड हला कि आनला प्राइमेज आसिला. प्राइमेज कम्स, सी प्राइमेज कम्स गोटे प्राइमर टाकू बाइंड कला. प्राइमर बाइंड कला पर कौन हबो पॉलीमरेज आसीबो. पॉलीमरेज विल कम एंड ऐड द न्यूक्लियोटाइड को ऐड करी करी से कौन करिवो ये करिवो. सेडा हला लेगिंग स्ट्रैंड. एड लीडिंग स्ट्रैंड देखिबा केमिति होची. सी कंटीन्यूअस सिंथेसिस होची. दिस इज द पेरेंट स्ट्रैंड and this is the leading strand. This is the DNA polymerase. See how the nucleotides at 3' OH and they are joined. Okay, now this is lagging strand, discontinuous synthesis. Okay, so this is the primer. Now behind the primer पछे पटे सब गोटे गोटे polynucleotide add हेकी ओकाजाकी fragment तयारी हला. Now another primer come. Okay, primase आसिला आउडे primer टे कला आउथरे again DNA polymerase 3 actually form polymerization adding nucleotides. So this is Okazaki fragment, this is another Okazaki fragment, like this another primer come and polymerase 3 polymerize the chain, Okazaki fragment again form. Okay, like this एमिति वे रे से कौन होची. एबे डीएनए पॉलीमरेज 1 आसिला. एडे कौन है रिपेयरिंग काम हला. रिपेयरिंग काम सम अनवांटेड डीएनए न्यूक्लियोटाइड थिला रिपेयरिंग हला. देन लाइगेज आसिला ताकू कौन कला से जॉइंट कला, ताकू गैप टाकू फिल अप कला. लाइगेशन रो काम कौन जॉइनिंग. तो डीएनए पॉलीमरेज 1 आसिला रिपेयरिंग काम कला, लाइगेज जॉइनिंग कला उथरे. This is how the total process is actually done. We will see another animation also to understand much more. See the double stranded DNA, it is in helical form. Helical form, these are the nucleotides. Unhelix hala, unhelix or unwind hala. These are the nucleotides. A double bond T, G triple bond C, which are complementary base pairing among each other. The two strands are there, which is one is 5 prime to 3 prime, okay, another is 3 prime to 5 prime. So both are anti-parallel to each other. Now the replication starts. This is a helicase. Here helicase is given. Helicase out topoisomerase रो complex है कि थाई सब बोले से माने पाखा पाखी सब बोले रहिबे सेति helicase लेखिला topoisomerase लेखिनी, but you just understand that topoisomerase is also there. Both are responsible for unhelix, unwind, and topoisomerase is responsible for releasing topological stress. So unwinding of DNA occurs. Now one replication fork moves in this direction, another that direction. The replication, the unwind hegala. Two different strand of DNA are unwinding. Then primase comes. Gote strand dekhiba example. Leading strand for example, primase come and primase form a primer. Primer taku tiyari karibo. 7 8 ki 10 ta nucleotide thibo. This is the primer. Which primer? RNA primer. Then after that, which come? Polymerase, DNA polymerase enzyme comes, and so we have seen the DNA polymerase comes and add all the free nucleotides, and the synthesis occurs from 5 prime to 3 prime direction. You see polymerase, this is the leading strand which is synthesizing, continuous strand from 5 prime to 3 prime direction. All the free nucleotides are actually added, okay. In this way the new strand is synthesized. And look into the lagging strand here. This is the primer, and 5 prime to 3 prime direction one fragment is formed. Gote fragment tiyari hala primer ta, eta okazaki fragment बोली कहले. तापर आउ गोटे आसीबो के प्राइमेज आसीबो, हेले प्राइमेज आसिला, प्राइमेज आसी गोटे प्राइमर टे तयारी कर देला. आमे पढुचे डिसकंटिन्यूअस स्ट्रैंड, लेगिंग स्ट्रैंड विषय रे. प्राइमेज पछरे आउ थरे पॉलीमरेज आसिला, डीएनए पॉलीमरेज. तापर दीज आर द फ्री न्यूक्लियोटाइड्स. सी 5 प्राइम टू 3 प्राइम डायरेक्शन रे आउ थरे सिंथेसिस अकर्स, सिंथेसिस हला. सेडा आउ गोटे ओकाजाकी फ्रैगमेंट. देन अगेन आउ गोटे प्राइमर आसिला, प्राइमेज आसिला, प्राइमर तयारी कला 8 to 10 nucleotides. Then DNA polymerase comes, and again the free nucleotides join together to form a Okazaki fragment. Immediately gote gote Okazaki si exonuclease will देखेला एठे. Exonuclease activity विषय मु कहली जोरा कि न्यूक्लिक एसिड को भांगीवार काम करे सी हला. हेले एडी किछरा आरएनए प्राइमर अछि ना, आरएनए प्राइमर रे सेठे यूरासिल मध्ये थाई पारे. आरएनए प्राइमर यूरासिल थाई पारे, से यूरासिल डा बाहर करियार अछि ना नै. तो सेथि पई दैट यूरासिल नीड टू बी रिमूव. द एक्सोन्यूक्लियस एक्टिविटी कहार अछि? डीएनए पॉलीमरेज 1 रो अछि जे कि रिपेयरिंग काम करे. सिमिली एक्सोन्यूक्लियस और डीएनए पॉलीमरेज 1 कहली एक्सोन्यूक्लियस एक्टिविटी पढ़ाई छी मु बिफोर क्लास रे, जस्ट अंडरस्टैंड. सो एक्सोन्यूक्लियस एक्टिविटी रे कोन हला? रॉन्ग न्यूक्लियोटाइड रिमूव हे गला बाय डीएनए पॉलीमरेज 1 एंड करेक्ट न्यूक्लियोटाइड एडेड. करेक्ट न्यूक्लियोटाइड ऐड हबो के करिवो? डीएनए पॉलीमरेज आसीबो करिवो. करेक्ट न्यूक्लियोटाइड को ऐड कला परे हला माने एटे जेहेतु मल्टीपल ब्रेकेज हला, गोटे न्यूक्लियोटाइड हेथिले डीएनए पॉलीमरेज 1 ही कर दे थांता. एटे डीएनए पॉलीमरेज 1 आसिला, सेडे न्यूक्लियस एक्टिविटी न्यूक्लियस आसी डिग्रेड कला और डीएनए पॉलीमरेज 1. एटे डीएनए पॉलीमरेज लिखिला बट दैट इज 1. जे कि नॉर्मली पॉलीमराइजेशन करछि दैट इज डीएनए पॉलीमरेज 3. सो डीएनए पॉलीमरेज 1 आसिला, देन ऐड कला, ऐड करिगी एति की फिल अप कर दला करेक्ट न्यूक्लियोटाइड को. देन लाइगेज आसीबो, जोड़ जोड़े गैप अछि रीजॉइनिंग करिवो, गैप गुडा को फिल अप करिवो. व्हाटएवर वेयरएवर द गैप्स आर देयर ओकाजाकी फ्रैगमेंट को जोड़िवो. प्लस एइडे मध्ये जोड़ जोड़ रिपेयर हेइथिवो सेइडे मध्ये डीएनए लाइगेज तार लाइगेशन करिवो. देन दीटा न्यू डीएनए स्ट्रैंड तयारी हला, अगेन दे फॉर्म हेलिक्स. सो दिस वे one DNA strand, one DNA double stranded DNA form two double stranded DNA. So this is the completion of DNA replication. So we have understood how DNA is actually replicating, okay, and the total process, the total enzymatic activity we have seen. तमर जदि आउरी भल भाव कर एनिमेशन वीडियो टाकू टिके रिपीटेडली देख सो दैट इमेजिनेशन टा पूरा परफेक्ट हो। थैंक यू वेरी मच। I hope this animation video will help you a lot to develop the proper concept in your mind. So watch this video class repeatedly. Thank you.